Okay, so this is part 2 of chapter 8. You guys watch this animation. And did I talk much about this slide? Do you guys remember where we stopped? Either way, I want to just say a couple more things. So looking at the diagram in the corner here, I want you guys to notice this green line. Do you guys see this green line that says interphase? So it's a little confusing because of all the different phases in this topic of cell division. It's kind of like the idea I think I mentioned with you guys before, of us through our life cycle. We go through different stages, or they say phases too, right? Like toddlers, or maybe the terrible twos, or things like that. So it's the same thing with the cell. So interphase is the phase that takes up 3 phases of the whole cell cycle. So you guys see what I'm talking about? So G1 is considered an interphase, S is considered an interphase, and G2 is also part of interphase. Now, interphase includes all these 3 phases because when you look at the slides under the microscope or you watch them doing what they do like you guys saw in the video, you can't tell the difference by looking at them. You can't tell the difference by what they're behaving. You can't see it. You can't see them growing up a little bit bigger. I guess maybe if you're doing very detailed observations you can, but not under killing the cells and staining them. You can't see it. So that's why interphase takes up all those, because all you see is a normal looking cell with a nice nucleus, and it's all blurry because it's full of the DNA strands. Then the cell actually starts to change what it looks like to us when it hits the M phase. And then that's when the DNA strands coil up and condense, and then we can see them as chromosomes. Okay, so we need to concentrate on the S phase in the beginning sections because this is very important on how DNA replicates. So it says during the S phase, the DNA of each chromosome replicates to form a new identical set of chromosomes. So I would underline identical. It's very important that you understand that mitosis is making exact copies of cells. So memorize it right now. S stands for what? Synthesis. Synthesis of what? DNA. It's making more DNA. Okay, just memorize it right now. The G1 means gap 1, like a period, a time period, a gap. But I also think of it, reminds me of growth. And that's what the cell's doing. In gap 1, it's growing, it's doing its job. If it gets the signal to reproduce, then it goes into S phase, copies its DNA. G2, another gap, is also about growth. Before the cell divides, it wants to copy everything it needs so that when it divides, it has enough of everything for 2 cells now, or at least getting it on its way. Because when the cell first divides in 2, it is smaller, and then it's back in G1, and it's going to grow some more. So it's this big circle of life. Okay, so then these are the 2 huge concepts here. Nuclear division is called what? Mitosis. Underline it, highlight it. So we actually name it mitosis because we're tracking the division of making 2 nuclei. And then cytokinesis, what's cyto mean?
Cytokinesis is division of the whole cell.
It is division of the whole cell. And do you guys remember what cyto stands for? Yeah, the cells. Okay, so that's how you can remember it. It's the actual splitting of the cell. So the plant's going to build a wall. The animal cells are going to pinch apart, okay? And it's actually called cleave. You might have noticed that on the video. Okay, then what do we call the cells? The original cell we call the parent cell, and it basically split into 2, and now we have 2 new, we call them daughter cells. Okay, so DNA is just amazing. it copies itself with the help of a couple enzymes. Let's see here. If you remember about DNA structure, and if you've forgotten this from the beginning, you're going to want to go back and review a little bit. There are bases. Do you guys remember these 4 different colors here in this particular model stand for the 4 bases? Do you guys remember any of them or what letters they're symbolized by? A's and T's and C's and G's. Okay, you got to memorize it right now. There is one more nucleotide, but DNA doesn't have it. It's an RNA. Do you guys remember that one? It's uracil, and it is ribonucleic acid is RNA. Deoxyribonucleic acid is DNA. Deoxyribonucleic acid, DNA. Okay? And yes, it's spiraled into this helix shape. Hydrogen bonds hold it in that spiral, but we won't worry about those too much. We need to worry more about the other hydrogen bonds. Do you guys remember that down the middle there, the bases are held together with hydrogen bonds? These are weak bonds, so the DNA can fairly easily open up. It needs to open up a lot. The DNA needs to split apart to reveal the code so that the proteins can be built off the code, as well as it needs to unzip to replicate, to copy itself. Oh, let's look at these side chains. Let's not forget that DNA on the sides is alternating sugar, phosphate, sugar, phosphate. Now everybody look at this very closely. The one on the right side over here, you see the phosphate connected to the sugar and its nitrogenous base. That's the whole nucleotide. And you could see how the different 4 nucleotides just keep repeating. Now notice, how is this side different than this side? How is this one different from that side? Everybody, eyes up here. Look, this is really important to notice. How is this side different than that side? What were you going to say? So they're opposite of each other, right? They're reversed. Do you guys see that? So this is known as anti-parallel. Again, it's not something you have to memorize, but I want you guys to see it because it'll make sense when you're making your DNA. DNA can only be made in one direction. And so it's going to go one direction on one side and the other direction on the other side. So they're parallel to each other, but they're going in opposite directions, and that's how they fit together. So a little bit closer look at the nitrogenous part, the rings, also known as bases. You have thymine, cytosine, adenine, and guanine. Notice that thymine and cytosine have one ring structure. This is a little more advanced. You don't need to memorize it, but that's a P. Pyrimidines, that's a chemistry cool thing, how they're naming and organizing and their descriptive names. Versus purines have the double ring. You do not need to remember that right now, but I just want to introduce it to you. And then think about it. So they're the same. So A's and T's bonded together, they're going to take up the same length, the space. Is that the same thing with cytosine and guanine? If there's a screw up, if there's a mutation, and a guanine gets bonded to an adenine, that's going to change the shape of the parallelness, right? It's going to puff out. If the 2 little ones bonded together, then it would be an indent. You guys follow me on that? Structure and function. Okay, a little bit more with the bonding. The backbone, the sugar phosphate backbone, is held together covalently, so it takes more energy to break that bond. Again, these ones are what types of bonds? The ones in the middle? Hydrogen bonds. Now, it's kind of interesting, they have realized that certain organisms like bacteria that live in extreme environments, like for example hot springs, they have more C's and G's in their code than A's and T's. Now look at the picture. Why would something that lives in a harsh, tough environment have more C's and G's in their code, in their DNA? So look at them carefully. What's the difference? Yeah, you see the C and G is held together with 3 hydrogen bonds. A and T is a little looser with only 2 bonds. So DNA is fragile because it needs to unzip, right? But things can cause it to unzip or fall apart, and then when it gets remade or put back together, it could have mutations. So things in harsher environments only make it if they're a little tougher, even their DNA. Okay, open your textbook to 217. I want you to see this nice 217. I want you to look at the figure there. Okay, so what's so cool is how DNA copies itself. And what it does is it does that by well, first it has to untwist, and enzymes help it untwist, so it gets flat in a part of it. Doesn't have to completely untwist. Then it unzips, right? The hydrogen bonds break, and then it can assemble the complementary side on both sides. So look at this closely, you guys. First, what are the green, blue, and goldish brown things? What are those circular blobs sort of things? Read them. What are they? What? They might have, okay, so they might have some RNA in them, but, so the overall, yeah, they're called that structure. They're enzymes, though. Do you guys see that? Okay, so what do enzymes do? They help with reactions, right? They put things together or break things apart. So in this case, the one that's actually making some breaking to get it unzipped, but the other one, and this is the main one that you need to memorize. What's the brownish one called? DNA polymerase. It's helping a reaction happen, and it's building. Do you guys see that? So it's building each side. And also notice you have a leading strand and a lagging strand because they have to be built in opposite directions. And what is so cool is how perfectly it will get copied using the template of the old DNA. So do you see in the new DNA, it's half old and half new? You guys following me? So this is beautiful. This is just amazing. Instead of using the DNA, and you don't rip this DNA model apart, and I give you all these pieces, and I say, here, build one just like this. It's going to take more time, and also, is it going to maybe have more mistakes? But since the pair bonding is fixed, A's only bond to T's and C's only bond to G's, you separate the two double-stranded into single-stranded sides. If there's an A there, what needs to attach to it? A T. If there's a C there, what needs to attach to it? G. Okay, so if you've forgotten the bonding, you need to write it down. I memorize it with the word at. A-T. A-T goes together. I know some people have memorized it with the capital C and G look similar, right? They're kind of shaped similar. I don't care how you memorize it, but you need to know it. So now, just because A's are only fitting to T's and C's are only fitting to G's, this thing gets replicated almost perfectly every single time. Okay? Where it starts to copy the DNA is just known as the replication origin. Not that important of a word. It also goes to tell you that with the chromosomes, in bacteria, there's only one, excuse the plural, right? One circular chromosome. So what happens is I did a little bubble here on the board because the DNA has to come apart, and then it only goes in one direction, and it starts in one spot until it goes all the way around. Then it'll end up with two circular pieces of double-stranded DNA. The eukaryotic is more efficient with the linear chromosomes, and it starts in several different spots along the chromosome, and so they could be replicating at the same time, and it'll just come apart quicker, and you'll end up with 2 strands. Little trivial, but kind of interesting. Now, the direction, like I said, is really important. So, because they're opposites, each side's going the opposite way, when it comes apart, the DNA polymerase only fits going one direction on it because of the way those sugar phosphates are shaped. The enzymes have to fit perfectly to their substrates. So on one side, it's no big deal. That's called the leading strand. So as the DNA is being unzipped, it keeps applying new nucleotides, the A's, T's, C's, and G's, and it's no big deal. As it opens, the thing just keeps chugging along and makes a continuous strand. Think about the opposite side, though. Now we have to go this way. We have to go the opposite way. So as the DNA comes apart, the DNA polymerase has to wait for the opening to get big enough to fit there. So it fits there, and it's got to go up and away from the opening, and then it's going to run in to one that it's already made. So if you look right here, so here it's being made, it's going to run into this one, and it can't keep applying. It has no place to go. It has to come off, and then it's left with some areas that didn't get made. And so no big deal. After there's some room there, another DNA polymerase, a special one, will come in and fill in these gaps in a different way. So these are called the lagging strand, and this is the leading strand. It's pretty amazing how it takes care of itself. And like I said before, this is called semi-conservative because half of the old DNA is being conserved. So it's like half conserved. So the DNA that you have in your cells today still could have the same piece of DNA that came from your mom's egg, really, or your dad's sperm, right? Because the old one just keeps getting copied, and half of it, and even you can think back even through generations, so it's pretty cool. So that piece of DNA you got from your mom could have come from the actual DNA that was in cells inside your grandma. And there's billions and billions of these nucleotides. So odds are you got some pretty old DNA someplace in your own chromosomes. So just kind of cool. Okay, the other amazing thing is how this very long, thin molecule gets packaged in your chromosomes. And then we can see them in the microscope, or on the video when you guys saw them. So look it up. I've actually challenged you guys. I did yesterday for some the A classes. Look it up and get some good analogies for this offline and share them on Edmodo. Do not share one that's already been shared, but there's all sorts of analogies. Like, for example, how long is your DNA in one cell if it was just the double helix? How long would it be if it was unwound and all 46 chromosomes? How long would it be if all your cells' DNA was stretched out? How long would it be if for all humans? Or what about all the life on Earth? So you can find these analogies. I'm pretty sure if I remember right, the one for one cell of a human DNA, I think it's a couple miles long. A couple miles long if that DNA was not wound up and circled up and folded up and all that inside every one of your cells. So if you want, look it up if you think that's kind of cool to check out and post them at Edmodo. Now, check out this slide. So here's the helix. This is what I'm talking about. And again, this is how it gets so small and how it stays fine inside your chromosome, in your nucleus. Even more important is how compact it gets during mitosis, because we need that long strand to be all put together, all folded up tight together so that it gets in the new cell. We can't miss any of the long strand. So it's crazy complex. So first thing, it's already spiraling. Then that piece right there, long piece, will start coiling also. Then it starts looping around, coiling around these histones that help make it even more compact. And that's those little red balls. Again, we still have a very thin thing at this point. So it coils up some more. Then that thing coils up some more until that whole line, however long it is, I don't know how long one of the chromosomes is, but if 46 of them make a couple miles, it's pretty long, right? And it's got all coiled up into that teeny, teeny little X that we can barely see, or we can see nicely with a microscope. So that's why chromosomes form, and that's why we only see the DNA when they're in chromosomes, when they're all supercoiled. Otherwise, it just looks like a blurry mesh. Okay, now even though DNA copies almost 100% perfectly all the time, 1 in a million on average, 1 in a million does get screwed up. Maybe a T was supposed to be there and a C was put there instead, or whatever. Okay, the wrong one bonded and it got slammed in there and it's maybe sticking out or sticking in. It's not right, but the DNA polymerase kept going and boom, we have a mutation. Okay, so a mutation is in the DNA. One of the A's, T's, C's, G's is in the wrong spot. That's all a mutation is. It'll make more sense next chapter of why this can be a problem. It's not always a problem. Most of your DNA does not code for a protein. It's not read. So it's like filler space, and it might have some other tasks that we're not aware of as of right now. But this makes us different from each other also. If it's in a protein that actually codes for if it's in a gene, that actually codes for a protein, it might make the protein different. And that also could give us variation. And it also could be bad, right? Harmful sometimes. So for example, sickle cell anemia, the protein in the blood, is misshaped for those people and they have this tough disease. That's one base pair difference, one mutation. We'll talk more about sickle cell in the future. But our hair color, eye color, our skin color, all these things that we can easily see are all because of different mutations that have accumulated. If the mutation made a change, like hair color, and it doesn't matter to the people, they can survive just as easily with their hair color, then it stays in the population. So back to this. If the mutation, the change in the DNA is not harmful, we call it a what? It's a silent mutation. They can be harmful, they can even be lethal. Also, the other thing to consider here is if we're just talking about some cells in our body, we have billions and billions of cells. So big deal, those cells divide. Those cells have a mutation, they die. No big deal, right? If you're talking about gametes, though, if it happened in your egg or your sperm cell and you pass it on to the next generation, then it can be more of a problem. Okay, we're almost done. Hang in here with me. Okay, so what causes mutations? Chemicals, different environmental factors, like smoking is just insane, what it's doing with your guys' lungs. And I mean, if you guys know people that are smoking, I mean, try and support them to stop because it'll catch up to them. As humans, we like to live right now, right? So long-term things, how we eat, how we take care of ourselves, lots of times we push off. And then it comes back to haunt us. But anyway, so these mutagens can mess up and make mutations where the DNA does not copy itself correctly and makes a screw up. What isn't so also amazing, back with the eukaryotes, if they have a replication problem, if they screwed up when they were copying themselves, like that 1 in a millionth time.
Although many cells in adult organisms are not actively dividing, they still need accurate instructions on how to function. Therefore, they need to detect and repair mutations introduced during replication or caused by environmental factors, such as mutagenic chemicals or radiation. Most mutations are known as mismatches because they consist of base pairs that cannot form hydrogen bonds, for example, adenine and cytosine. The process by which these mutations are repaired is known as excision repair. First, an enzyme recognizes the mismatch and binds to the DNA.
See the greens? Oh, stopped.
It then breaks the sugar-phosphate bonds of the mismatched section and removes the damaged or mutant DNA. Next, DNA polymerase fills in the deleted DNA sequence. Finally, another enzyme forms sugar phosphate bonds between the replacement piece and its neighboring nucleotides.
Okay, so then even that 1 in a million base pairs can get screwed up. With eukaryotes, they have this proofreading mechanism. So then it's closer, believe it or not, to like 1 in a billion. So the mutations are very rare. Okay, so this last part I'm going to go through really quickly. This is what the movie explained really well to you guys. So again, we're just finished up in the S phase. We have all our DNA copied. And now the chromosome is not linear. It is an X shape. The two exact copies of DNA are being held together at the centromere. And then they are each known as chromatids. And they're exact copies, so we call them sister chromatids. And there's just another picture of that. Oops. Okay, so also you remember in the video, there can be a mutation of the chromosomes where the chromosomes don't get ripped apart, right? Or separated correctly. Do you guys remember that in the video? One of the cells, one side, right? One didn't get ripped apart, so it has 2 linear chromosomes. The other one didn't get any chromosomes. That's called non-disjunction. So sometimes then we can get cells that have the wrong number of chromosomes. So that's neuploid. Later on, we'll talk more about haploid cells and diploid cells. It all has to do with the number of chromosomes. Haploid have half the chromosomes. Diploid have 2 of every chromosome like us. You got 1 from your mom and 1 from your dad. Okay, so here's a picture of interphase. Look at this closely. You cannot see the cell membrane. It's just too light. But what you're seeing here is the nucleus. You can even see nucleoli. There's 2 in some onion cells. You can see the fibers growing. So that's an interphase. Now, when we get to prophase, you guys look carefully, those chromosomes are coiling. The DNA, called chromatin, the DNA strings are coiling. So now we get to see blank space. You see the white there because they're getting all organized. This is the first part of prophase. The movie broke down prophase into prophase and prometaphase. I'm not overly concerned about it, but this is prometaphase down here. The nuclear envelope is gone. The chromosomes are attaching to those spindle fibers. You can faintly see the cell membrane in that side. The kinetochores is another vocab word. It's actually the structure that's on each chromosome or chromatid that is at the centromere. Little trivial. Okay, now check it out. Metaphase. How are we going to remember this? M. The chromosomes are lining up in the what? Middle. Okay, so this is metaphase. This is just starting. Okay, then we hit anaphase. Now the chromosomes are being ripped apart. They're getting back to their linear, correct shape. I memorized this with, they kind of like a V or an A without the cross, right? And that's because that spindle fiber is dragging it by the centromere, and then the legs of the chromosome are just lagging behind, right? They're falling behind. Okay, telophase is what we drew with Alexis, except we did it with plants. This is animal, and you could see the dye with the fibers. The chromosomes are getting on together. They're starting to relax and uncoil. You could see cytokinesis, the pinching together in that picture. Okay, so kind of let's quiz yourself real quick. You guys, eyes up here. So, you guys see this one is anaphase. This one is what? Telophase or telophase. How do I know? What's the difference? Do you see the plate forming? What's forming right here? The cell wall. Awesome. Okay, here's metaphase. You have to remember they're not all lining up perfectly for your photograph when we do this or when we're looking. So that's metaphase. That's metaphase. What do you think that is? Metaphase. What do you think that is? Metaphase. What about this one? Okay, now, these are prophase. You could see the white, the chromosomes are forming, the nuclear membrane is breaking down. This is interphase, 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 interphase. Just, just blobs. So what do you guys think this one is? Prophase. You can see the chromosomes forming, and the membrane is breaking down. Now here's some animals. Metaphase, prophase, anaphase, interphase, interphase. Blow up, same thing. And just a repeat. Okay, what's this one? Yep, telophase. I can see the line. I can see the wall forming. What's this one? This is just prophase. The stuff is still, it's just the nucleus is breaking down. They're coiling. Okay, what's this one? Everybody, what is it? Metaphase. What's this one? Anaphase. What's this one? Prophase. Okay, we stopped. Made it.